In the last section, we were talking about the difference between gram-positive and gram-negative bacteria. The reason why this is so important is that different antibiotics work on the different types of bacteria differently. So depending on what kind of infection you have, you would be prescribed a different type of antibiotic. Today's antibiotics can sometimes be classified as broad spectrum. Broad spectrum antibiotics can be used fairly equally on both gram-positives and gram-negatives, which means it's not absolutely necessary to diagnose a patient before prescribing them. We're going to talk later about how broad spectrum antibiotics, though easier for doctors and hospitals, are actually causing an increase in antibiotic resistance in our world today. The difference between gram-positive and gram-negative cells isn't just in their cell envelope. There are molecules in the cell envelope of gram-negative cells, particularly in the outer membrane of the gram-negative cells, that can be classified as endotoxins. Endotoxins are molecules that are released only when the cell dies in lysis. In this case, those endotoxins, when released, can lead to damage of host cell tissues. So in trying to fight and kill those gram-negative bacterial infections, you could end up leading to damage to your own cells. Gram-positive cells do not have this outer membrane, and without that outer membrane, they do not have those endotoxins. Instead, some of them can produce exotoxins. Exotoxins are toxins or molecules that are secreted by the bacteria. And this secretion is done only when the bacteria are alive. So while the bacteria are causing an infection, they are releasing these exotoxins to make the disease in you even worse. This happens with strep throat, particularly when the Streptococcus bacteria grows on the surface of your pharynx or respiratory tract. By using antibiotics to kill these cells, you would be stopping this process. That brings us to a discussion about the cell membrane. The function of the cell membrane is to regulate movement of molecules into and out of the cell. It's to keep the good molecules in, like DNA, RNA, proteins, and food, and keeping bad molecules out, like antibiotics, disinfectants, and other things. The bacterial plasma membrane is thus referred to as being selectively permeable, meaning some molecules are able to easily move in and out, while others cannot. Molecules that can easily move across the membrane are gases like carbon dioxide and oxygen. Even water can move across the membrane using osmosis. On the other hand, molecules like food or exotoxins all cannot be transported across the membrane unless the cell has ways of pumping them across. The plasma membrane is not unique to bacterial cells. In fact, all cells have a plasma membrane. There is no cell on Earth that doesn't have one. And nearly all these cells have a plasma membrane made of the same thing, phospholipids. The plasma membrane is made of fatty molecules, whereas the cell wall and the capsule are both made of polysaccharides or carbohydrates. Going further into the inside of the cell, the first thing that you would encounter would be the cytoplasm. The cytoplasm is the fluid-filled space inside the bacterial cell. Again, all cells have these, but the cytoplasm of bacteria do not have any internal structure to them. They contain a whole bunch of different molecules floating around freely within the cytoplasm. The cytoplasm is mostly water, but it also contains other things like DNA. When the cell goes to divide, it must divide the DNA into two different halves of the cell, and then divide the cell in half by joining together the cell membrane and then growing a cell wall in between. So the first step of this process, known as binary fission, is to replicate the DNA of the cell, the chromosome. After DNA replication, those two pieces of DNA are sectioned into different halves of the cell, one on each side. A fission ring forms, creating a separation between the two halves in the form of a cell membrane. After the cell membrane forms, a bacterial cell wall forms on top of that, separating the two. This is called the septum. Once the septum forms, you have two individual separate bacterial cells. And that essentially is binary fission. The DNA that's found inside bacteria is always going to be a bacterial chromosome. Every single cell on Earth has to have DNA. And in bacteria, that DNA, as you can see from this picture, is circular. It's contained within a circle. There is no beginning or end to the DNA molecule. That circular molecule, called the bacterial chromosome, is often looped around on itself so that it can fold and fit inside a single bacterial cell. That section where the bacterial DNA is housed is not surrounded by a membrane, and so it's not a nucleus. However, as you can see from this picture, it is compartmentalized to a portion of the cell. That compartment or region where the DNA is located is called the nucleoid. The DNA found in the bacterial chromosome is usually circular, and there's usually just one of them per cell. This is different from, say, your cells. In human cells, we have our DNA in 46 pieces. So we have 46 chromosomes. In addition, our chromosomes, as we'll learn later, are linear. So there's a beginning and an end to them. Usually, scientists draw these as X's, if you've ever seen these in books or classes that you've taken. The bacterial chromosome isn't always just the only DNA inside the cell. Sometimes the cell can also contain a second piece of DNA known as the plasmid. Plasmids, unlike chromosomes, are not required as part of the cell's survival, but instead can provide an advantage to the cell. Plasmids are also almost always circular, but are much, much smaller than the bacterial chromosome itself. That's because the bacterial chromosome contains thousands of genes required for bacterial cell survival, but the plasmid usually just contains a few to a few dozen genes that can provide an advantage to the cell. Plasmids are really important for lots of bacteria that have them because they can encode for things like antibiotic resistance, heavy metal resistance, or genes that cause disease, known as virulence genes. In addition, scientists use bacterial plasmids quite frequently in genetic engineering studies. Also, unlike bacterial chromosomes, plasmids can be shared from one bacterial cell to another. They do this through a protein bridge that extends from the bacterial cell. This protein bridge is what's called a pilus. The bacterial cells can then send a portion of their DNA, the plasmid DNA, through that pilus bridge. The first cell that's donating the plasmid retains a copy of the plasmid DNA, and the second cell receives the copy. And so, in the end, both cells end up with the plasmid, and both cells can share that advantageous DNA with other bacterial cells. Another really interesting and important structure found in some bacterial cells is the endospore. Endospores are extremely hardy. They are highly resistant to heat, drying, radiation, and various chemical compounds and disinfectants. Endospores have been shown to survive for thousands of years and can even survive the vacuum of space. There are only 3 genera that we know of on Earth that can form endospores. Your book talks about 2 of them. One of these is Bacillus, and the other is Clostridium. The third one your book doesn't mention is the Sporosarcina. Sporosarcina are not causal agents of disease, and this is why your book doesn't mention them. But the Bacillus genus and the Clostridium genus are major risk factors for some human diseases. The endospores are essentially a dormant state. So when the bacterial cell encounters an environment that is not advantageous for its survival, it will end up going through sporulation and form endospores, where they'll protect a single cell encased in a very thick layer, allowing it to survive for thousands of years until a good environment comes again. When the good environment comes back, the endospores can essentially germinate, much like a seed. And when the cell comes out of the endospore state and starts growing and dividing again and is no longer dormant, it's referred to as the vegetative state. Vegetative cells are really easy to kill, whereas, as I mentioned, endospores are very difficult. Endospores forming bacteria include Bacillus anthracis, which causes anthrax, Bacillus cereus, which causes foodborne illness, Clostridium tetani, which causes tetanus, Clostridium botulinum, which causes botulism, and Clostridium perfringens, which causes gas gangrene, a common disease found in the Civil War. Now, in addition to these structures, some bacteria also have extracellular appendages, or little tails or things that can come out from outside the cell. One of the more common of these is the flagella. Flagella allow the bacterial cells to swim. Flagella are made of a protein called flagellin, easy to remember. And these flagella from bacteria spin much like a propeller on a boat or a plane, allowing the bacterial cells to swim through fluid. The flagella are often many times longer than the single length of a bacterial cell, but unfortunately are so thin that they cannot be viewed under the light microscope unless they have a special stain attached to them. As you can see from this picture, different bacterial cells can have different numbers and arrangements of flagella. Don't worry about knowing these different types for the quiz. Flagella are often attached to the bacterial cell by a structure known as the basal body. Bacteria can control the speed and rotation of the flagella using the basal body. They would do this sometimes in response to a chemical and can swim toward or away from chemicals. Of course, this is how bacteria can swim toward food or away from toxins and poisons. Other bacteria can connect their flagella and basal bodies to the detection of light energy, that's called phototaxis, or oxygen in aerotaxis. For bacteria that move toward or away from chemicals, this is called chemotaxis. And all of these are regulated by movement of the flagella. There are 2 other structures that can cause bacterial cells to not swim, but instead stick. Fimbriae are one of these, and pili are another. Fimbriae exist all over the surface of the bacterial cell, like hairs covering the surface of the cell. Pili, on the other hand, come in just 1 or a few per cell. They can get up to hundreds, but most often they're in just a few per cell. While the Fimbriae just help the cell stick, pili often help cells stick when they want to share plasmid DNA with other cells in a process known as conjugation. Pili are made of pillin. Another easy to remember protein name.